All right, guys, so citric acid cycle this week. It's going to be awesome. Let's get to it. So, to just orient ourselves a little bit, we are coming out of what I kind of call the acetyl-CoA step. Now, it's really more often considered like the end step of glycolysis, but it's a really important step, so sometimes we separate it out, which I like to separate it out. We talked about how it's a really big complex, three giant protein complexes with all these domains, the pyruvate dehydrogenase complex. It's really important that you remember what was going on there because we're actually going to use an almost identical complex about halfway through the citric acid cycle. So anyway, to orient ourselves, remember we're taking a pyruvate and we pulled one of the carbon dioxides off of it. And in so doing, so we're oxidizing pyruvate, and in so doing, we've pulled energy and electrons away and put them onto NAD+, making NADH. And that's then going to then go to the electron transport chain, which we're going to talk about next week. And we're left with our acetyl group, which is those two carbons, plus our coenzyme A that's holding on to it with its sulfur. Now, to kind of give you a visualization of what's going on, I want to cut to another North Dakota State University virtual cell video that's just going to kind of get us right on the same page.
Cell. Let's take a look at the major steps of the citric acid cycle. In the first step of the cycle, a 2-carbon molecule and a 4-carbon molecule are combined to form a 6-carbon molecule. That 6-carbon molecule undergoes multiple biochemical changes during the cycle, and at the end, the original 4-carbon molecule is produced. Each time a carbon molecule loses one carbon, a carbon dioxide is released. So 2 carbon dioxides are formed during the conversion of the 6-carbon molecule back to the 4-carbon molecule. So where does the 2-carbon molecule come from to begin the citric acid cycle? This molecule, acetyl-CoA, is made from pyruvate. Pyruvate is a product of glycolysis, the other major ATP-producing cycle in the cell. Pyruvate is transported into the mitochondrial matrix, where it is oxidized to acetyl-CoA by pyruvate dehydrogenase. This step produces one NADH and one carbon dioxide for each of the 2 pyruvate molecules made from glucose. The acetyl-CoAs are the starting point for the citric acid cycle. In the citric acid cycle, the acetyl group from acetyl-CoA is transferred to oxaloacetate to form citrate. 4 different enzymatic reactions then lead to the formation of succinate. These steps produce 2 carbon dioxides, 2 NADHs, and 1 ATP. Succinate is then recycled back to oxaloacetate through 3 more reactions. These steps produce FADH2 and 1 more NADH. All of the energy molecules made in the citric acid cycle, along with those produced by glycolysis, are essential for fully functioning cells, as errors in these pathways can lead to life-threatening diseases.
That gives you a better just overall synopsis of what we're doing. And so we're just going to kind of talk about how we get there. So let's start with our first reaction, which is reaction number 1, citrate synthase. And so for each reaction, I try and put the enzyme at the top, and then a little bit of description of what's going on. And so in this case, we're having a condensation reaction. So because we're condensing these two things out, we're going to take our acetyl group, we're going to take that coenzyme A off, okay, because we're kind of using it as a carrier to bring the acetyl group over to citrate. So citrate came from, oxaloacetate and acetyl-CoA are going to be combined to make citrate. So oxaloacetate is what's going to come through here. And so I'll highlight what's happening with this nice blue box. You're going to watch this blue box go around. And so we're going to condense our oxaloacetate with our acetyl group to form citrate. And so what's kind of cool, I've just got lots of fun facts about these kinds of things. So the active site of citrate synthase is really interesting. So it binds oxaloacetate first, which makes perfect sense. It's coming from the rest of the reaction, right? So you have an oxaloacetate, it binds to the active site. That opens up the active region of the active site for the acetyl-CoA to actually come in and bind. And we're going to hydrolyze that coenzyme A. So it's a thioester. So remember that coenzyme A has it's like this big old honking molecule with a sulfur on the end. And sulfurs allow us, as you recall, to form special kinds of covalent bonds called thiols. Thiols are really cool. So sulfur has an extra layer in its outer layer of its electron orbitals, which allows us to pull sulfurs and the bonds, the covalent bonds between sulfur and another molecule much more easily. We can separate them than other kinds of covalent bonds, like between carbon and an oxygen, for example. And so when we actually hydrolyze it, which we're going to talk about in a second, it's super exergonic, which is awesome. It's going to drive, it's one of the most exergonic things in this entire system, and it's going to allow us to drive this entire process, okay, which is very, very cool. So to understand what's going on, I want to remind you of reaction 9 of glycolysis. So in reaction 9, we learn that if we form, I'm just going to jump ahead to the molecule that's actually there. So this is phosphoenol pyruvate, which here's your pyruvate, okay, 1, 2, 3 carbons, okay. We're going to lose this when we go through from pyruvate to acetyl-CoA. This is the carbon dioxide that we actually lose in that process. And so to take phosphoenol pyruvate and pull that phosphate off, we generate the enol form. And so the enol form is when you have a carbon-carbon double bond like this, okay? This oxygen is highly electronegative, and so it would prefer to have this double bond between the oxygen and the carbon, not between the two carbons. So this is the not preferred form. And so when we have an enol form like that, that's what that's called, an enol, we jump back and forth between our enol and enolate and then into our keto form. And this is the preferred form. And so this pressure of wanting to go into the keto form is what we're going to take advantage of during these kinds of reactions, okay? And so we're going to take advantage of that, and we're going to generate a carbon-carbon double bond, which will then allow us to drive the reaction forward. Let's get into this. So we're back in the citric acid cycle. We've got our oxaloacetate here, okay? So this is this molecule right here. It's coming from over here. And we've got our acetyl-CoA. There's our acetyl group, 1, 2 carbons attached to a sulfur with a coenzyme A. So that's the coenzyme A structure. But they always show the sulfur just to show that thiol. Now, during this first step, what's going to happen is we need to form that enol form. Okay, so if I just back up, right, we need to form a carbon-carbon double bond, okay, because that's not preferred, but that's kind of going to drive us forward, right? So two things happen. We need a carbon-carbon double bond. So see, there's a carbon-carbon double bond here. And so what we need to do is get the double bond off of this, between this oxygen and this carbon to go away, because we need to form a carbon-carbon double bond. So if we have one carbon with 1, 2, 3, 4, 5 bonds, well, that doesn't make sense. So we have to take one of these bonds of oxygen off just for a second, okay? And so we're going to do that. So we attach one hydrogen from this histidine, which is hanging out right here, which is convenient, to this oxygen. Okay, so it's going to hold on to it here. Okay, so we got rid of that double bond. Sweet. And then we actually need to form that double bond. And so this hydrogen, which is attached to this carbon here, is going to attach to this aspartate, this aspartic acid up here on top. And so that's going to allow us to form a double bond between the two carbons. So there's our enol form, okay? And now that is not preferred, right? We know that our enol form is definitely not the preferred form. It wants to jump back to our keto form. And so what we're going to do is take advantage of that. So we're going to take one of these double bonds right here between the carbons. We're going to flip that out and bond it over here to our oxaloacetate. So we're going to actually form our new citrate molecule, which is an acetyl group plus our oxaloacetate. And we need to reform our double bond between the oxygen. So that's switching from our enol form to our keto form. And that's what we see here. So we now have our carbonyl here. So there's our double bonded oxygen to our carbon. Sweet. So that's a keto form. And then we've taken, I'm just going to kind of back up. So look, there's our double bond here. And we're going to take one of these and flip it out and attach it to this carbon right here, smack dab in the middle of oxaloacetate, which is awesome. Now, that's going to allow us to then use water to take water and throw it onto the coenzyme A with that sulfur, and we're able to sort of like kind of just replace this acetyl group with the water. Okay, and so then the coenzyme A goes away, it gets recycled. We're going to encounter it again very, very soon, and actually like 3 different steps, so don't miss it for too long. It'll come back, it's okay. And then we're left with oxaloacetate, so check that out. That is a cool looking molecule. Now, something I always want you to remember is that when we're actually doing these reactions, the active sites of these amino acids is the active sites of these enzymes is really, really important, and the actual structure of the amino acids is really, really important. The location of these amino acids is really important. And so what you can usually do, like if you're trying to do some research on an active site, how they figure these things out is they would know the DNA sequence, and then they could change one sequence, so they could know, like, okay, if we mutate histidine 320, so this is the 320th amino acid, if we mutate that from a histidine to glutamine or a glycine or something, just we change it to something different, then we lose the ability. And you could tell which ability is lost. Like maybe you only end up with Citrate complexes because you're not being balanced out by this histidine here, or whatever. It doesn't really matter. But that's just really important. The actual residues is really, really important. Let's jump into our second reaction, which is sort of a two-parter, but we can kind of lump it all together. It's a two-parter. I'll explain why in a second. So what we're going to do is we need to perform an isomerization, okay, which we've encountered before. Glycolysis, we have some isomerizations. Basically, we just need to move things around. So we need to change citrate to an isocitrate. So citrate itself is a tertiary alcohol, but those are a pain in the butt. So we need to switch to a secondary alcohol, which is called isocitrate. Before we get into that, this is endergonic, but it's reversible. But the trick here of the citric acid cycle, it's kind of similar to some of the steps in glycolysis in that reaction number 1 and reaction number 3, which we're going to talk about next, are super duper exergonic. So I don't know if I mentioned that before. So this is negative 32 kilojoules per mole for that first reaction we just talked about, okay? And then the next one is also super endergonic, or exergonic, I'm sorry. And so we have a really, really strong push from reaction 1 and a really strong pull from reaction 3. And that's what really gets us through this a-KETOGLUTARATE reaction, this isomerization. It's really interesting. So the active site of a-KETOGLUTARATE is really, its structure is really weird. We're going to talk about that. And so we have a positive 13.3, but we're able to actually defy that because of how negative our other ones are. And so if we actually look at citrate, so here's the structure of citrate. I added this nice circle here. It's a tertiary alcohol. What does that mean? Well, that means your alcohol group or your hydroxyl group here is attached to a carbon. Okay, so there's the carbon it's attached to. That's bonded to 3 other carbons. So 1, 2, 3. So that makes this a tertiary carbon. That is a tertiary carbon, which makes this a tertiary alcohol. Okay, tertiary alcohols, you have to break, if you wanted to oxidize them, you'd have to break carbon-carbon bonds, which is a huge pain in the butt. And so what we want to do is make it a secondary alcohol. Those are much easier to react. And so it's easier for us to switch from a citrate to an isocitrate than to actually try and break a citrate down in a variety of different ways, okay? So we're just kind of doing this step to make further steps easier, okay? Just like any kind of other isomerization that we do. And so we need to move this around. Now, it would be nice if you could say, like, okay, well, I'm just going to take this alcohol group, and I'm going to take this hydrogen, and I'm just going to pull them each off, and then stick them onto different spots, right? So I want my alcohol on this carbon here, and a secondary alcohol, and I want my hydrogen on this one. Well, it's not that simple. Basically, what we do is we pull off this alcohol group and this hydrogen, which forms water, and then we're going to re-put water back on here, but we're just going to use it to put it in different spots. And so the end product, yes, it's this alcohol group is now on the secondary carbon and then this hydrogen here. But just to kind of understand the intermediate step is different. We form a carbon-carbon double bond here. So basically you're taking the half of this bond and half of this bond and sticking it right here. So they kind of fold over and they fold over here. So we have a double-double bond. And then we're going to fold this back out and just reattach it to an alcohol group for this carbon and hydrogen for this carbon. And that's called isocitrate. Now, the enzyme that does this, a-KETOGLUTARATE, is really interesting. And so if you actually look at its active site, it's very, very cool. And so here we have our citrate, and they're showing arrows of how it's about to become an isocitrate. And I just want you to see there's some really interesting things going on. Lots and lots of hydrogen bonding is occurring because there's all these nice oxygens, and we got all these alcohol groups hanging out. And the active site actually has something that we're going to see in the future, which is called, I mention it here, an iron sulfate or an iron sulfur center. Okay, we're going to encounter a few of those, a lot of iron sulfur compounds when we actually get to next week's talk, which is the electron transport chain. So I kind of wanted to just expose you a little bit to this. But it turns out you can make weird things called iron sulfur centers. That should freak you out. It looks crazy, right? But remember, we see iron and we see sulfur a lot in biological systems. And so in this case, what we're going to do is we're going to use this to sort of form bonds and stabilize the actual structure of citrate and allow us to, you sort of hold it in place so that you can pull that water off and then put it back on in a different spot, okay? And that's what this is for. We see sulfur a lot. I mean, think about all the uses that we have for sulfurs, like with the coenzymes, okay? So it comes up a lot, and so that's really important to see. This happens to be held by cysteines as well. So there's like disulfide bonds that actually hold this on there. Pretty cool. So let's go on to reaction number 3. So the enzyme here is isocitrate. Okay, that's what we just made, right? So we're going to take isocitrate, dehydrogenase. So whenever you see a dehydrogenase, that is awesome. So what we're going to do is we're going to use dehydrogenase to dehydrogenate something. We're going to take a hydrogen off, and in the context of the citric acid cycle, we're going to take that hydrogen and the energy and electrons with it when we break that bond and stick it onto NAD plus to make NADH, which, if you'll recall, NADH is one of our electron carriers that we're going to take into the electron transport chain to put a hydrogen on the other side of the membrane and allow it to pass back through to generate that hydrogen gradient, which is awesome. So this is really, really great. So this is one of the first steps where we're going to actually generate something cool. So this isocitrate dehydrogenase is the enzyme, and it does 2 things. So it's going to reduce NAD+. Okay, so we're going to take that hydrogen, take the electrons, bond, and stick that hydrogen onto NAD+ to make NADH. So we're reducing NADH, i.e. we're oxidizing isocitrate dehydrogenase. Okay, and in the process, we're actually going to decarboxylate isocitrate. And so we're left with isocitrate. So you'll notice the structure of isocitrate. It has, let me back up. So here's our isocitrate, and we've got these nice CO2 molecules sticking on these edges here. And so what we're going to be able to do is pull off one of those carbon dioxides. It's going to be awesome. So that's called decarboxylation. That's why you breathe out carbon dioxide. This enzyme is one of the reasons you breathe out carbon dioxide. That's so cool. Now, the intermediate is a ketone, and it's unstable. Okay, we're going to see an enol form. It's going to be awesome. So recall, when we have those enols, we can take advantage of that. Okay, so what we're going to do is we're going to kind of follow the enol, all right? I'm going to talk about NAD plus and NADPH in a second. So here is what's happening. So there, see that CO2 right here. Okay, so this should look super familiar. This should look just like this guy, right? So there's our CO2. We're actually about to lose this, and we've got our alcohol on our secondary carbon and our hydrogen here. Okay, so there it is. There's our alcohol and our secondary carbon, hydrogen, and there's our CO2. And now what we're going to do is we're going to rip off this hydrogen, and we're going to stick it onto NAD plus to make NADH. Okay, and then, so that's awesome. So we've reduced NAD plus. And then we're going to pull off this carbon dioxide here, okay? So that's pretty cool. So we're left with α-KETOGLUTARATE. The intermediate form is called oxalosuccinate. Sorry, so many words. Oxalosuccinate. In between the oxalosuccinate and the α-KETOGLUTARATE, oh man, you form an enol intermediate. And we know enols are not the preferred form. We prefer the keto form, hence why this is α-KETOGLUTARATE, okay? And so we're taking advantage of that. So I'm not going to go back over everything that we talked about, but that's how we're going to have a negative delta G. Once again, it's because we're going back and forth between that enol and the keto form. Now, something needs to be said first about this enzyme. So there are a couple of different forms of this enzyme. Basically, they're all the same, except they have an interchangeable region. So you would make one protein with one domain that can take a hydrogen and stick it onto an NAD+, or you can swap out a domain and stick it onto an NADP+. Okay. Now, we're not going to go into this too deep. You'll see NADPH versus NADH. Okay, so NAD plus becomes NADH. NADP plus becomes NADPH. Okay, so you'll see NADPH if you've taken botany. That's what gets generated during photosynthesis. But mammals and all kinds of other creatures use NADPH. We just tend to use it for anabolic processes as opposed to catabolic processes. So in usually what we're talking about is we make NADH that goes to the electron transport chain. It's a continuation of the oxidation of our carbon molecule, okay, of our glucose, for instance. NADPH would be, we could send that to like the pentose phosphate pathway if we wanted to generate other kinds of molecules, okay? So it tends to be used more, at least in mammalian systems, for for anabolic processes, NADPH. NADH is generally for more catabolic processes. Something we need to talk about, just to give you a little tidbit. So isocitrate dehydrogenase, it's also called IDH. There are a few different isoforms of IDH, and they do different things. Like I said, some can make NADPH, some can make NADH, but they can have mutations, and this is one of the more common ones that you encounter in certain cancers, especially brain cancers. And so what they do is, instead of making α-KETOGLUTARATE, they can accidentally keep going and convert the α-KETOGLUTARATE to 2-hydroxyglutarate, okay, or 2-HG. What 2-HG is really bad. So 2-HG is what we call an oncometabolite, and so it's made during a metabolic process, hence it's a metabolite, and it's onco meaning cancer. And so what it can do is it can go and it can have a few different effects. Ignoring the prolyl hydroxylases here, which it just changes your collagen and all kinds of crazy stuff. But the genetic landscape part here is the part I want to talk about. And basically, what it can do is it can inhibit demethylation, okay, which is really confusing. So when your DNA, well, your histones and your chromatin, so when your chromatin has lots of methyl groups on it, it's really, really tight and it can't be transcribed, okay? And so what happens, if you demethylate it, then it will open up, right? Now, what can happen is certain cancers can inhibit that demethylation. So they're keeping it from opening up. They're keeping it nice and tight. So it keeps that chromatin methylated, which decreases transcription. And what they can do is they can do that to regions that have tumor suppressor genes. And so you can't transcribe the tumor suppressor genes, thus you can't make tumor suppressor proteins like, oh man, we've talked about them before, like P53, BRCA, there's a ton of different ones. And so that's why this is really, really bad. And so if we take a look here, basically what's happening is we're generating α-KETOGLUTARATE, that's fine. But what happens when we have our mutants is they keep going. So they'll take that α-KETOGLUTARATE and they'll take the carbonyl here next to the carboxyl group, and they will turn it into an alcohol group, which it can no longer go into the correct system. So it stops the citric acid cycle and then has all these other different effects. And so that's really, really interesting. And so that's isocitrate dehydrogenase. And so we're moving on to reaction number 4, which is a really, really cool reaction. So we're going to take that α-KETOGLUTARATE, okay, which now has 5 carbons, right? Because we lost, we had our 6-carbon citrate, we moved things around, and we lost one of those CO2s with isocitrate dehydrogenase. And now we're left with α-KETOGLUTARATE. So we have 5 carbons. And what we're going to do is we're going to send it into a complex called the α-KETOGLUTARATE, okay, dehydrogenase complex. Now that should sound super familiar because we used a pyruvate dehydrogenase complex to go from pyruvate to acetyl-CoA. And so now we're going to go from α-KETOGLUTARATE to Succinyl-CoA. It's almost identical. It's literally almost the exact same thing. It's going to be awesome. And so we get the exact same stuff, right? So we have that coenzyme A and we get an NADH, just like we did when we went from pyruvate to acetyl-CoA, right? We generate 1 carbon dioxide and 1 NADH and our acetyl-CoA. And so in this case, we're going to take our α-KETOGLUTARATE. We're going to generate 1 NADH and 1 CO2. And so we're left with a 4-carbon molecule. Now, sometimes, and the little video that I showed you, they talk about this is kind of the halfway point, okay? So we're reaching the halfway point of the citric acid cycle, because what we're going to do is we're going to take our Succinyl-CoA and sort of restore it back to oxaloacetate. We're not going to lose any more carbon dioxides, okay? And so that's where we're at. This is going to be awesome. So we lose, where we had that carbon dioxide up here, we're going to replace that with our coenzyme A, which has that nice sulfur thiol group right there. So it's literally the exact same thing that we saw with when we oxidized pyruvate and used the pyruvate dehydrogenase complex. And so this is a slide from that slide set where we took pyruvate, okay, there's our pyruvate, we get it into the mitochondrial matrix, and we're going to send it through this 3-enzyme complex, right? We have our E1 where we attach it on there and we lose the CO2, that's that one's job. And then we send it over to this crazy dihydrolipomide acetyltransferase group, the E2 complex. which has all these sulfurs, right? We use that to kind of hold on to things, and we use that to actually move it over into the next step. And then what we can do as that's happening in between is we get a couple hydrogens on those sulfurs, which allows us to then reduce FAD to make FADH2. Okay, so we've seen this before. The complex that actually performs what we're talking about now, the alpha-ketoglutarate dehydrogenase complex, is almost the same. I mean, for all intents and purposes, it's exactly the same. It has 3 complexes. It has all the exact same stuff. Let me show you. I know the quality of this is really low. I am so sorry. But it's like one of the better diagrams, so I just went with it, okay? So there's our alpha-ketoglutarate, right? 4 carbons, 1, 2, 3, 4, 5. Sorry, 5 carbons, duh. We're about to lose this carbon dioxide. So we do that. That's the first step, right? Which, just like before, that was our first step, right? We lose that carbon dioxide in the pyruvate dehydrogenase complex. And this is TPP, same thing. See, TPP, TPP. We lose that carbon dioxide. So we move from our E1 complex, and we're going to move into our E2 complex, which has that 2 sulfur spot, which is going to hold on to our succinyl group. So we've lost our carbon dioxide from our alpha-ketoglutarate, and we're going to make our succinyl, which we're going to now call our Succinyl-CoA, because we're going to attach it to a coenzyme A, and that's what happens in the E2 complex. So we can see over here, we're going to take our now succinyl group, and we're going to attach it to one of the sulfurs on in the E2 complex, which then we kind of swap it for the sulfur in the coenzyme A. Basically, what we're left with is Succinyl-CoA, okay, so this bottom right corner guy, and then we're left with 2 hydrogens attached to these sulfurs, which we can then send over to the E3 complex, and the E3 complex's job, just like here, so we've got 2 hydrogens, 1, 2. The E3's job is to take FAD, and it's to reduce FAD, right? So reduction is the gain of electrons, and so we're going to throw some electrons onto FAD to make FADH2, which we can then use to reduce NADH, okay? And so that's the job of E3 here. So you can see I kind of colored each hydrogen so you can keep track of them. And so we're going to take those hydrogens and stick them onto NAD and make NADH, which is awesome. That goes to the electron transport chain. NADH is the preferred form, whereas FADH2 we use if we have to. And we're going to talk about that in a minute. So let's move on to reaction number 5. And we're going to see these last few reactions are actually going to go a little bit faster because what we're sort of doing is we're restoring back to oxaloacetate. And so we're just going to kind of follow that process. So we've got our Succinyl-CoA, right? So that came out of that giant complex. So we're stuck onto a coenzyme A with that sulfur. And we're going to use Succinyl-CoA synthetase. And we're going to do something really, really cool. We're going to do substrate level phosphorylation, just like we did in glycolysis. So if you recall, in glycolysis, we input 2 ATP, right, to stick those phosphates onto our glucose. And then we are able to get 4 of those out. And so in that process is substrate level phosphorylation, as opposed to oxidative phosphorylation, which is when we're using the electron transport chain to actually generate ATP. So in this case, we're using substrate level. So what we need to do is we're going to use the power of our enzyme's active site to take an inorganic phosphate, okay, and we're going to stick it onto a nucleotide diphosphate. So either like ADP is what we usually think of, but we can also use GDP. CDP and UDP tend not to exist in biological systems, though they can exist. We use them for other things. You can generate them synthetically as like chemical or like chemicals or weapons, like anti-cancer drugs, things like that. But for the most part, ATP and GTP are what we want to make because we can use those for different things. And so we're going to use substrate level phosphorylation to actually make those. Okay. So Succinyl-CoA itself as a molecule has a ton of energy. It's super exergonic when we actually break it. And so what we're going to do is we're going to take advantage of the fact that it's so exergonic to separate succinyl from that coenzyme A. And because it's so exergonic, we can use that to make ATP or GTP. Because we know actually making ATP is really, it's super endergonic, right? It's like 32, it's positive 32 for our delta G. And so a kilojoules per mole. That's usually what I have. When I'm talking about like our delta Gs, I'm doing kilojoules per mole because that's what Leninger, the textbook, actually uses. You can go other units as well, but kilojoules per mole is what I usually use. And so this is so exergonic we can use it to make ATP. And so what we're going to do, it overall is an exergonic process, right? So if I take 32 minus 36, I get 4 left over, right? So it's -4, so this process actually keeps on going. And what's really interesting is when we actually form this, which I'm going to show you how that looks, when you form either GTP or ATP, so this can be confusing. So in some citric acid cycle pictures, you'll see ATP listed as a product of this process. Sometimes you'll see GTP listed. Now, I was confused when I first learned this, and so I finally did some digging and figured out. So in mammals, basically, we can use Succinyl-CoA synthetase if you're in the brain, the heart, or in the muscle, okay, in skeletal muscle, or heart muscle, or whatever. You generally want ATP because that's what you're going to quickly use to do those processes. Now, if you're in like the kidney or the liver and you need to do more signaling molecule production, you need to make some GTPases. You might need GTP to just for a couple other processes, not necessarily for energetic processes like you would use ATP for, like for kinases, but for more signaling pathways is what you tend to use GTP for more. And so that's where you would see that more in like the kidney or the liver, okay? And so that's where you see that separation. And so it depends on the cell that you're looking at. But what's really, really important is inside of a cell, you can use a nucleoside diphosphate kinase, okay, so it's a kinase, right? Kinases phosphorylate things. And what that can do is it can take an ATP and make a GTP, or vice versa. It can take a GTP and make an ATP. And that is energetically free. Okay, so it's a 0 delta G. And so you can go back and forth between ATP and GTP however you want. So in the end, it doesn't really matter. You're doing substrate level phosphorylation and making something. And so what we're going to do is, there's our Succinyl-CoA, right? There's 1, 2, 3, 4 carbons. Awesome. There's our sulfur up here. And so what we're going to do is we need to stick an inorganic phosphate onto it, which we're eventually going to pull off. And we're going to lose that coenzyme A, and we're left with a kind of nice little equal little molecule here. We've got CO2 with 2 carbons right in between, and we're going to generate some a nucleoside triphosphate. So we're going to take our Succinyl-CoA, and how do we actually generate the GTP? Well, that's a good question. So we need to get our inorganic phosphate into the active site of our enzyme. And so that's Succinyl-CoA synthetase. So that's this guy here, okay? And we're going to attach an inorganic phosphate onto this carbon here because we're able to, it's pretty reactive, okay? So we can kind of flip this little bond out here and react it to our phosphate, okay? And using the enzyme's active site, which is going to use some positive charges, probably magnesium, and it's going to use those positive charges to actually pull those electrons away and allow the reaction to occur. And so we end up with this succinyl phosphate, okay? And so succinyl phosphate is kind of weird. It only exists for a little while because what we're going to do is we're going to take that phosphate, so it's stuck on there, and we're going to attach it to the histidine residue on the Succinyl-CoA synthetase, okay? So we're still in the active site. There's that histidine. We're going to stick that phosphate onto it, and then we're going to lose the succinate. So that's what we're left with. So there's our succinate. That falls off. But that phosphate is still attached to the histidine residue in that enzyme, which we know enzymes have to restore their active sites to their original state, right? That's super important. And so that phosphate does not like being stuck on that histidine. It just hates it. And so what we can do is we can send a GDP or an ADP nearby, and it will react. That last phosphate can react very readily to that phosphate. This bond falls back, the electrons fall back onto the histidine, and we are left with a triphosphate. So either an adenosine triphosphate or a guanine triphosphate. Sweet. That is substrate level phosphorylation. Move-ins, because these last couple steps are actually a little shorter and more dense, more condensed, sorry. So what we're going to do is we're going to take our succinate, and we're going to use Succinate Dehydrogenase, which, hey, always remember, when you hear dehydrogenase, that means we're going to either be making NADH or FADH2. Awesome. Those are the molecules that we're trying to make, because when we make those, we can send those to the electron transport chain and use those to make ATP. So those are awesome. Those are some electron carriers that we want to make. They're like big batteries. If ATP is a small battery, then these are like big batteries. Okay, and so we're going to take succinate, which has 4 carbons, right? I've talked about that already, And basically what we're going to do, remember, this whole process, we're trying to oxidize, right? When we oxidize, we're going to break bonds, remove electrons, and stick them onto these electron carriers, which can then take them to the electron transport chain, which we can use to move that energy and move hydrogens to one side of a membrane and generate a concentration gradient. And so what we're going to do, anytime we can do that, we're going to do it, okay? Now, it's not always, just because we're breaking a bond doesn't mean we're necessarily removing something. And so basically what we're going to do is we're going to take our succinate and we're going to rearrange and break some bonds, move some things around to pull some hydrogens off and generate a couple intermediate electron carriers, while all in the process of heading towards oxaloacetate. So that's the two processes that are happening simultaneously. We need to look like oxaloacetate. Awesome, we're going to do that. And in the same process, we're going to move things around, further oxidize our molecule, and generate a couple of these electron carriers, okay? So the whole point is to store energy and electrons and make them do work, okay? I want you to keep it in your heads that we're oxidizing glucose, right? That's usually what we're starting with. I'm going to talk a little bit in a little while about, it doesn't have to be glucose, but basically we're usually going to start with glucose. And then we're going to generate these other compounds, okay? So we're oxidizing glucose in our carbon compounds and reducing other things. So our electron carriers, i.e. ATP we just made, NADH we've made already, and we're about to make FADH2, okay? Now NAD plus and FAD are super powerful oxidizing agents, okay? So they really like to be reduced, okay? They're oxidizing agents, thus they like to be reduced. And so because they're so powerful, they're very exergonic. And so actually forming them is decently easy. It's spontaneous. It will happen as long as we sort of provide a space for it to happen with our active site. Now, as a general rule, when we break our carbon-oxygen bonds, like we lose our carbon dioxides, that's a very exergonic, or yeah, it's very exergonic, and we get a lot of energy out of that. And so we can throw a hydrogen onto an NAD plus and make NADH, okay? So we can reduce NAD. Now, with FAD, we don't get as much energy out, but we can get some energy. And so FAD we use to sort of hold a little bit of energy, not as much as from a carbon-oxygen bond. And so we switch to FAD to hold the carbon-carbon bonds that we can break and move. And so it is harder to oxidize FADH2 in the electron transport chain. And so in general, we only get, it depends on where you read, between 1.5 and 2 ATP per FADH2 versus you get 2.5 to 3, usually I just say 3 and 2, for NADH. But hey, it's better than nothing. We're going to use it. And what's really interesting is that this actual process, so this process in the electron transport, in the citric acid cycle occurs right next to the electron transport chain. So we're going to see this next week. So this is one of the transport complexes in the electron transport chain, which is really, really cool. And so this actually occurs right there. So here's tricarboxylic acid cycle. That's citric acid cycle. It's the same thing. That's what we've been talking about. We make our succinate, okay? And we need to go from succinate to fumarate. And in so doing, we're going to take 2 of these hydrogens off. We're going to form a double bonded carbon, which, remember, we don't love double bonded carbons. That's generally a weird thing to make. But in so doing, we're going to take each of these hydrogens and we're going to stick them onto FAD to make FADH2. So we have now reduced FADH2. Awesome. I want to talk a little bit about FAD, okay? So the structure of FAD is really interesting. I know the quality of this isn't great, but this is what's happening. So this is our succinate, okay? And we're going to take one of these hydrogens, this one, and we're going to stick it onto a nitrogen over here. And we're going to take another hydrogen, and we're going to stick it onto another nitrogen in the FAD itself, which I'm going to show you another picture of this, so don't worry about this. But that leaves us, so this bond kind of flips down here, this bond's going to flip up here, and so we form a double bond between these 2 carbons. Okay, so let's look at the actual structure of FAD. So it's flavin adenine diphosphate is what the actual name of this molecule is. That's what the F stands for, is flavin, which you may have heard before. Okay, so that's your B vitamins. I'm going to talk about that in a second. But here, there's the actual structure. So this is the really, really important part. So here's the adenine diphosphate. Okay, so there's the adenine. Okay, so that should look super similar to ATP, right? So here's this right here is ADP. But instead of, so we take ADP, adenine diphosphate, and then we're going to stick on a riboflavin, whatever you want to call it, riboflavin 5' monophosphate. And so we're going to stick that riboflavin thing, molecule, onto our ADP, which is really, really cool. So that's another connection to make, is that we talk about ATP all the time, going from ADP to ATP. But what's really nice is it's a really great energy carrier molecule. And so we can take ADP, stick this crazy thing onto it, and use it as another kind of energy carrier molecule, which is super cool. And so there's those two hydrogens that are going to come off of succinate. We generate fumarate, and then we're left with an FAD, okay, there's the AD, adenine diphosphate, there's the F, the flavin, and it's FADH2, right? We've added these two hydrogens on here. They were hidden underneath, but I made them red so that they stand out. So that's those two hydrogens there, which is awesome. And so we're going to send those hydrogens to the other side of the mitochondrial membrane, and we can let them pass back through and generate ATP. So what is this? Well, how do you actually make FAD? So it's flavin adenine dinucleotide, and it comes from B2 vitamin, okay, which is riboflavin. So you basically just throw a phosphate on the end. So you take this dude, you throw a phosphate on the end, and then you attach it to AMP, basically, sort of. We're going to see it a bunch. Why do I keep talking about this? Because we're going to encounter flavins a bunch. And so next week, we're going to talk more about flavins. And I always just like showing this. So if that sounds familiar, so riboflavin is the precursor, that's B2 vitamin, is called riboflavin. That's the precursor to NA or FAD, right? Well, where have I seen that before? Well, if you're a kid and you're staring, OMG, at the back of your cereal box, there's riboflavin. They put that in your cereal. Look at that right there. A lot of these vitamins are actually part of these metabolic processes. So either glycolysis or the citric acid cycle. They're these intermediates. This is why the US government requires manufacturers to put riboflavin into this cereal. It's supplementing your citric acid cycle. That's so crazy. Isn't that crazy? I think it's crazy. Awesome. So moving on, we are almost done, okay? So there's less reactions in the citric acid cycle than the glycolysis cycle. Is it a cycle? Not really. So we're going to take fumarate hydratase. It's a lot of times it's called fumarase. They just shortened it down. But I actually like the term fumarate because that's the precursor, right? We're going to take fumarate, and it's a hydratase. So we're actually going to hydrate the carbon-carbon double bond. And so we're sort of restoring, we're going to take this carbon-carbon double bond, because in general, you don't like carbon-carbon double bonds. You want that double bond to either not be between two carbons or to between, we're going to kind of break it and put it between a carbon and an oxygen. So that's usually what we're trying to do, okay? And you can look at oxaloacetate, which that's the next process is going to go from malate to oxaloacetate. So we're almost there. And so what we need to do is we need to end up with a carbon-oxygen double bond for oxaloacetate. And so what we need to do is kind of set that up. And so when we go from fumarate to malate, what we're doing is we're just going to take water. Okay, this is an irreversible process, by the way. We're going to take water and take one of those hydrogens. Here. Why am I talking about it? We're going to take this double bond, and we're going to have one swing out here. So this carbon's going to get a hydrogen. Gets a hydrogen. And then this one's going to swing out, and it gets this hydroxyl group. Okay, so we've stuck an alcohol onto that. And then this bond here is going to become a double bond. So you can see we got this carbonyl group here. We're going to swing, this oxygen is going to end up with a double bond here. Okay, so that's what we're going to do, but we have to set it up. Okay, now this is exergonic, so it likes to happen, but that's really all that happens. It's kind of a boring one. The actual enzyme structure, I don't know, it's slightly more interesting, but there's honestly not a ton of stuff out there about this. These last few processes, I don't know, it's pretty straightforward. You're just sticking a water onto fumarate. I don't know. It's not that crazy. So we're going to take malate, which looks like that, and this needs to be a double bond, right? So that's what we're going to do. So we need to generate oxaloacetate, and so we need to pull the hydroxyl group off of that, and we're going to take that basically we're going to take this hydrogen, and we're going to stick it onto NAD plus and make NADH, okay? Which it doesn't like to happen. So this is going to go onto NAD plus, okay? Cool. So that's what we're going to do. Ends up with a double bond on oxygen. So we end up with a double bond here, and that becomes NADH, which then goes to the electron transport chain. Awesome. Why does this even happen? Because to actually go from malate to oxaloacetate is not preferential. It's a very, very endergonic. It's a massive endergonic, actually. Well, why do we do that? Well, if you think way back in this talk a while ago, I said that reaction 1 and reaction 3 are very special. They have super duper negative, i.e. they're super exergonic reactions. And so citrate synthase will grab oxaloacetate like it's nobody's business and pull it and make it into citrate, okay? It's crazy good at it. In fact, the intramitochondrial concentration of oxaloacetate is on the order of 10 to the minus 6 molar, okay? That's crazy low. That's how fast. Citrate synthase is basically just sitting around waiting to find oxaloacetate. As soon as it sees it, it grabs it, sends it through the process. And so that is one of those processes by which we've talked about this before with enzymes, right? You can push a reaction to happen and you can pull a reaction to happen. And this is one of those cases where citrate synthase is pulling oxaloacetate, i.e. the product of malate dehydrogenase, okay? That's the enzyme. I forgot to put that there. So, okay, it says enzyme malate dehydrogenase. So, but it's a dehydrogenase. Remember what I said, if you hear dehydrogenase, that means you're making NADH or FADH2. And in this case, we get one last chance to make NADH, which is awesome. It's gonna go, see that arrow? Sweet. We're gonna send that to the electron transport chain. So just to kind of show you what's happening. So this, I got this from Wilmington Corporation. They sell stuff like chemicals and stuff. So there you go, Worthington. I couldn't get rid of the watermark, but it's like the super high quality picture. That's why it takes so long for this slide to actually load the first time I do it. So there is our malate, okay? And so we've got our oxygen, our alcohol group stuck to that carbon, and we want to make this a double bond, right? So there, it's our oxygen with a double bond, so that's what we're going to end up with. Okay, that's oxaloacetate, which we're going to send with acetyl-CoA to make citrate. And so this is NAD, and this is NADH. And you can see some similarities here, right? Here's the nicotinamide, and then it's ADP, just like FAD, right? So the structures are super duper similar. So that should make you, should give you some comfort in your life. And so right here we have our NAD. This becomes NADH. What's kind of confusing is that to go from NAD plus to NADH, yes, there are two hydrogens here, but you're just moving them from different spots. Plus it's got a hydronium ion. Don't worry about it. You're going from NAD plus to NADH, basically, okay? And then we're going to take that hydrogen and put it on the other side of the electron transport chain, which allows us to generate more electrons. It's going to be great. That's next week. So I found this diagram. It's from Leninger, I think. But it's these last couple processes. So going from, remember we went from Succinyl-CoA to succinate, and then we're restoring oxaloacetate. And so just to reiterate, there's kind of two steps to this process. Some people don't like it that way. Some people prefer to just kind of memorize all the steps as they're happening. But I kind of like this way, splitting it right down the middle, where you have the one side, which is generally getting your carbon dioxides out, and then you have your other side, which is going from succinate to oxaloacetate, sort of restoring the whole process. Okay, and so this is just one way to look at it. Basically, all these other diagrams that I've had, like this one, this is, see, it wouldn't load. This is this exact same one, I just cropped it. So anyway, so that's just a nice little summary there. We're making some FADH2, and we're going to take a water and make an NADH. It's going to be great, and we end up with oxaloacetate. Now, something important as we kind of take a little breather here and we just sort of step back and look at this whole thing, because I went through a lot of details. There's a lot of enzymes. Thank you, Khan Academy, for this picture. It's simplified. We have 6 carbons, and we're going to lose 2 of them and make some NADHs. We end up with a GTP or an ATP, and we're going to end up with an FADH2 and an NADH and end up with a 4-carbon oxaloacetate. And then the process starts all over again. Now, if you recall, if we took one glucose molecule, we get 2 acetyl-CoAs. So you go through this whole process twice, okay, which generates 8 NADHs, 2 FADH2s, and 2 ATP, okay? So like this one. So if we add all of that up, we end up with 32 ATP, and then if we include from acetyl-CoA and glycolysis, the NADHs from that as well, then we end up with 40 ATP minus the 2 that we had to use to break glucose. And so that's where we actually end up with our 38 ATP, okay? Now, what I wanted to do was jump back over to sort of give you guys another visualization, just like I did with glycolysis. to cut to North Dakota State University. They have another, it's still a relatively short video, but just kind of visualize physically what's happening in 3D space, this whole process. So let's cut to that video.
In the first step of the cycle, an enzyme called Citrate Synthase joins the 2-carbon acetyl group from Acetyl-CoA with the 4-carbon oxaloacetate to form a 6-carbon citrate. In step 2, an enzyme called α-KETOGLUTARATE converts citrate into isocitrate. Next, an Isocitrate Dehydrogenase enzyme oxidizes isocitrate, a 6-carbon molecule, into a 5-carbon α-KETOGLUTARATE. The carbon that was lost is released as carbon dioxide, and 1 NADH is also formed. The carbon dioxide that is released was originally part of oxaloacetate and not acetyl-CoA. In the fourth step, an enzyme called α-KETOGLUTARATE dehydrogenase converts alpha-ketoglutarate into a 4-carbon Succinyl-CoA. Similar to step 3, this reaction produces 1 carbon dioxide and 1 NADH. In step 5, a Succinyl-CoA synthetase enzyme converts Succinyl-CoA into succinate. This produces GTP, which is converted to ATP. In step 6, an enzyme called Succinate Dehydrogenase converts succinate into fumarate. This step makes 1 FADH2. In step 7, a fumarate HYDRATASE enzyme then converts fumarate into malate. In the final step of the citric acid cycle, a malate dehydrogenase enzyme converts malate back to oxaloacetate. Like all steps involving a dehydrogenase, a coenzyme is produced. Here, it is NADH. The oxaloacetate that was regenerated through the citric acid cycle is now ready to join with another acetyl group and begin the cycle a second time. For every 1 glucose that is broken down through glycolysis, 2 pyruvates will be produced. These 2 pyruvates will produce 2 acetyl-CoAs. So for every 1 glucose, 2 acetyl-CoAs will be made and 2 turns of the citric acid cycle will occur. This means each product of the cycle must be doubled. A total of 4 carbon dioxides, 6 NADHs, 2 FADH2s, and 2 ATPs are made through the citric acid cycle. NADH and FADH2 are electron carriers that can produce more ATP later in aerobic respiration.
The citric acid cycle. Now, there are 2 more things I wanted to talk about, okay? Which each of those have subcategories, so 2 more things, more like 10, but whatever. So, how do we control this? When we talked about glycolysis, same thing came up. You can control different enzymes. There are 3 key ones that we actually control in the citric acid cycle. So just like in glycolysis, there are 3 major enzymes. I think reaction 1, 3, and 9, 10, I can't remember, are the key controllers for glycolysis. In the citric acid cycle, it's these 3. So citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex, okay? And so those all have really negative delta Gs, okay? So they really want to happen, right? They're very spontaneous, okay? And so they're really easy to drive forward. And so that's why we control those, okay? So if you control the ones that are super exergonic, that's an easier spot to control. If it's endergonic, then what's the point of controlling it? It's not spontaneous. And so these are the ones that we control that are spontaneous, especially citrate synthase. I mean, we just talked for a little bit about how oxaloacetate is so rapidly pulled away from this process and sent into citrate. And so citrate synthase is really, really good at that. The other thing that controls is generally the ratio of your NAD plus to your NADH. That's really, really important. So you need a ton of NAD plus if you actually want to go through this process. So just think about that. So we think a lot of times about we're making NADH in the citric acid cycle. Okay, that's awesome. What if there's no NAD plus around? Well, what are you supposed to do then? If there's no NAD plus, how can I make NADH? That's a great question. And so if you're not, if your electron transport chain is not functioning for whatever reason, maybe you don't have your terminal electron acceptor, like oxygen, or maybe you're dead, which means you don't have oxygen. And so you're not able to take NADHs and turn them into NAD pluses, okay? And in the process, you're also not making ATP, of course, but the whole point is you're not taking NADH and making NAD plus. And so if you don't have NAD plus around, that is a substrate that you need for a bunch of these processes, right? At least 3 of them, plus you need FAD for your Succinate Dehydrogenase. And so if you don't have those around, that's a really good way to control this whole process. Another interesting side note, Different cell types will do different things, just as if this wasn't already confusing enough. So like in the liver, for example. So the liver can have super variable citrate levels. So citrate is one strong way that actually goes back into glycolysis. So if you go back and look at the control levels for glycolysis, now that you've watched this, it will actually make more sense because those control diagrams show citrate blocking phosphofructokinase. Okay. In glycolysis, and that would slow things down. So if you have a bunch of citrate, which is right here, right? So if you're making citrate, you're making a bunch of this, then it's not being pulled away, okay? So if I see a bunch of this, so much so that it can go back and affect my phosphofructokinase, that means there's a ton of this sitting around. It's not getting pulled through this process. Therefore, you're probably not, making a lot of NADH and pulling it into the electron transport chain. So it's just kind of sitting around. And so if you have a bunch of citrate sitting around, then you want to turn off your glycolysis, because why would you make this acetyl-CoA? Why would you even go through this process if there's a bunch of this already here? So it kind of backs up, sort of. But that it physically is blocking phosphofructokinase's abilities. Now, heart cells, okay, are unable to export citrate into the cytoplasm. So this is what's kind of tricky about this. So in your liver cells, they can transport citrate out of the mitochondria and into the cytoplasm where glycolysis occurs. Your heart cannot do that, which kind of makes sense. You don't ever want your heart cells to sort of be blocked from undergoing glycolysis, okay? They're that important. We can deal with excess glucose in different ways. You don't want your heart cells stopping doing that. Okay, and so why in the world would you send a bunch of citrate into the cytoplasm if it could possibly inhibit phosphofructokinase? Because I don't want that to happen. I want my heart cell always making ATP. And so we don't make a citrate blocker, or I'm sorry, a citrate transporter, really. We don't make a citrate transporter in heart cells, which is really interesting. And now here's one other thing to think about with that. How do we control that? Because every cell in your body has the genes for every protein that you can possibly make. So your heart cells can make the citrate transporter that could send it into the cytoplasm, but they don't. Oh, interesting. Must be some transcription factor epigenetic crazy stuff going on there. Now, the very last thing I wanted to talk about are a couple of terms that get thrown around, okay? This is anaplerotic versus cataplerotic, and anabolism versus catabolism. Now, we've talked about anabolism and catabolism before. Now, anabolism is building things, okay? So it's an anabolic process. Catabolism is breaking things down, right? So if it's catabolic, you're breaking things down. So like glycolysis is generally a catabolic process that breaks down glucose into pyruvate, okay? So that is catabolic. Now, the citric acid cycle is considered an amphipathic process, okay? Because you're breaking things down, right? It's catabolic, right? We're taking citrate and pulling carbon dioxides off, and we're generating energy, right? We generate NADH. But you're also making compounds, okay? Especially think about going from like malate to oxaloacetate and restoring it. So we're restoring those compounds. That in and of itself is an anabolic process. Important to note. Well, let me back up a little bit. So we're generating lots of different molecules, okay? So oxaloacetate being one of them. If you look at this chart, there are tons of intermediate molecules that we can take out of the citric acid cycle and send them to different places, okay? I.e., if you want to make like fatty acids, you use citrate as the precursor to make fatty acids. You use oxaloacetate to make a bunch of different amino acids. There's a ton of these different ones. You can use α-KETOGLUTARATE to make glutamate, which you can then use to make other different amino acids. And so the citric acid cycle, the tricarboxylic acid cycle, the Krebs cycle, it's amphipathic. It doesn't have to be taking citrate and breaking it down to generate energy. That's not its only job. So that would be the catabolic side of it, but it's also anabolic in that it's making these compounds that might go do something else. Now, those intermediates are what are referred to as either anaplerotic or cataplerotic. And so as you generate these intermediates, so let's say you made the α-KETOGLUTARATE, okay? If I make α-KETOGLUTARATE and I pull it out to make a glutamate, okay, that's called a cataplerotic process or a cataplerotic reaction. If I took glutamate and converted it into α-KETOGLUTARATE and sent it back into the tricarboxylic acid cycle or the citric acid cycle. That's called anaplerotic, which it feels kind of backwards to me, but anyway, that's the terms that are used. So if I'm pulling things out of this, I'm catabolizing that product and sending it into a different reaction. And if I'm sending something in, that's anaplerotic. So it kind of makes sense. Like I'm sending it into here to be generated into something else, so that's an anaplerotic. If I'm pulling it out, it's sort of being catabolized for something else. I have one more chart too that just shows where different amino acids come in or leave, which is just kind of a fun little chart there. Anyway, that's the citric acid cycle. It's a lot, I know, but hopefully it was a little clearer now than it used to be. And we're going to tackle the electron transport chain next time, and it's going to be really awesome because we're going to tie everything together with a nice bow.